oogenesis. It is the process of formation of the functional haploid ova from the diploid germinal cells in the ovary or female gonad. Early stages of oogenesis occur by the time female fetus is only 25 weeks old. At this time, all the oogonia have been formed and entered into the primary oocyte stage. Multiplication phase. Certain cells of the germinal epithelium ovary which are larger than the other functional germs they undergo repeated mitotic division to form diploid oogonia. Some 2 million oogonia gamete mother cells are formed in each fertile ovary. The oogonia form egg tube of P-plugger which grow into the cortex and give rise to a multicellular mass called egg nest. Growth phase. One oogonium of the egg nest grows inside and function as primary oocyte. The other cells of egg nest form an epithelial covering ovary. The same is called follicular set or epithelium. The in-seated primary oocyte is called primary follicles. A number of these follicles degenerate between the birth and puberty. At puberty, an ovary contains 60,000 to 80,000 primary follicles. Maturation it begins in the pubertal stage of females, but it is interrupted quite early. The primary oocyte begins meiosis 1, but the division is arrested in the reproductive stage of prophase 1. It is in this phase that ovarian follicles containing the oocyte occur in the coitus and remain so till the female attains puberty. After attainment of puberty, follicles start and their content resume development, usually one at a time and once a month. Follicles differentiate into layer of granulation, or another layer called theca develops from the cortex. With the formation of the theca, primary follicle transform into the secondary follicle. Theca differentiate into two layers, theca externa and theca interna. Granulosa cells secrete fluid that cause development of the cavity or antrum around the primary oocyte. The stage is tertiary follicle. Primary oocyte grows further and meiosis 1 it produces a large secondary oocyte and a small polar body. Both are haploid. The polar body has a very small amount of cytoplasm, but the whole chromosome set. In human females, the polar body does not divide further. The bulk of nutrient-rich cytoplasm is retained in the secondary oocyte. The follicle grows to maximum size and is called Graafian follicle. Zona pellucida develop around secondary oocyte. The secondary oocyte proceeds with meiosis 2, but the division gets arrested in metaphase 2 stage due to accumulation of metaphase promoting factor in this stage of oocyte that ovum is set during ovulation. It passes into oviduct where in the ampulla part cell cycle will resume only after the entry of sperm. It triggers the breakdown of APF and promotes synthesis of anaphase.